I am Dr. Josephinol, Assistant Professor, Department of Biochemistry. So in this video, we are going to see about the gene transfer methods. So first we should know what is gene transfer. So gene transfer is nothing but the insertion of genetic information into the host cells in the form of DNA is known as gene transfer. So gene transfer can be done by several methods. Then objectives of this unit is, so after completing this video, you will be able to answer the questions of what is gene transfer, and by using electroporation techniques, how we can introduce the foreign gene into the host cells, and by using the microinjection method, how we can introduce the foreign gene into the host cells. Then gene transfer method, there are several methods are available for these techniques. Here gene transfer methods. So gene transfer techniques can be divided into two types: indirect gene transfer methods and another one direct gene transfer methods. The indirect gene transfer methods include the agrobacterium mediated gene transfers, and then direct gene transfer methods again divided into two types: physical methods and chemical methods. Then the physical methods include electroporation, microinjection, macroinjection, then biolistics and liposome mediated gene transfer techniques. Then similarly, chemical methods also include a number of techniques. So here we are going to see about only the physical methods. Then artificial method of gene transfer. Here the exogenous gene is transferred or introduced into the host cells by using either chemical methods or physical methods. So first one, electroporation. So electroporation is a process used to introduce the foreign gene into the host cells. So here we are using electric shock to introduce the foreign gene into the host cells. Here we are applying high voltage electric pulses which create a pores on the plasma cell membrane. So through these pores, the foreign gene enter inside the host cells. Then inside the host cells, what will happen? The foreign gene bind with the host genome to form a new cells. The main principle involved in this technique is the high voltage electric pulses can cause the plasma cell membrane to fuse. Then this is a diagram for electroporations. So here you can see that here this is the cell that which is surrounded by plasma cell membrane. So here we are applying electric shock, high voltage electric shock, which creates a pores on the cell membranes. You can see here a pores is created here. So through this pores, foreign gene enter inside the host cells. Then it bind with the host genome to form a new cells. Okay. So next one, microinjection. So microinjection is a technique used to deliver the foreign gene into the host cells. Here for cultivated cells, the transfer of DNA by microinjection is commonly employed. So here the microinjection is a technique used to deliver the foreign gene into the host cells by using a glass micropipette. Then the tip of the micropipette is about 0.5 mm diameter, which resembles the injection needle. Then the transfer of DNA by microinjection is done under powerful microscope. Then the empty needle is removed after the tip of the micropipette is inserted into the cell membrane. Then this is diagram for micropipette. So this is micropipette. This is micropipette. So through this micropipette, we are introducing the foreign gene into the host cells. So this is cell which is holding in the holding pipette. Okay. Holding pipette. So through this micropipette, we are introducing or deliver the foreign gene into the host cells. Then summary. So both the methods are used to deliver the foreign gene into the host cells through the cell membrane. Then these are the possible questions in this unit. Here you can see the various types of questions. So these are also questions. Then references for this unit. Then thank you.